Okay, so this is part 2. The whole reason why I took you through all of this terminology is so that we can apply it to a comparison between mitosis and meiosis. Now, when we do this, it's gonna make everything make sense. All right. So stay with me. So as you can see here, you see a cell with 4 chromosomes. 1, 2, 3, 4. And sorry about the resolution. Okay. So let's just fill in all the numbers. So that's what I want you to do. For every cell, I want you to fill in the numbers. N equals 4. And the ploidy number is 2. Why is it 2? Because for the number 1s, you have a white and a black one. For the number 2s, you have a white and a black one, okay? 4 divided by 2 is 2. That means, that makes sense because N represents the number of kinds of chromosomes. You have 2 kinds here. You have 1s and you have 2s. Right? Remember, kinds refers to the numbers of the chromosomes, not the ploidy number, not the sets, not the colors. All right? I'm talking about the numbers here. C is equal to 1 because they're figure 8s. S phase happens. Next, after S phase happens, you duplicate the amounts of chromosomes, okay? So that's why the copy number becomes 2 down here. How many total chromosomes do you see now? 1, 2, 3, 4. So this number on the right has to be 4. Okay? How many sets of chromosomes do you have? Well, let's look at the number 1s. You have a black number 1 and a white number 1. You have a black number 2 and a white number 2. You have 2 sets for each kind of chromosome. Therefore, it's diploid, or number 2. Okay, let's do algebra. 4 divided by 2. 2. N equals 2 means there are 2 kinds of chromosomes here. You have 1s and 2s. Regardless of the color, regardless of the set, okay? So you have 1s and you have 2s. You don't have 3s, you have only 1s and 2s. That's why this is N equals 2. Alright? Now, what's going to happen is these will line up in a straight line in a single file, and they're going to divide at the centromere. And whenever you divide at the centromere, you're going to decrease the copy number by half. So let's look at these 2 cells, the 2 daughter cells. What's going to happen is you have 1, 2, 3, 4. So you have 4 chromosomes here, and you're going to be diploid still, because for the number 1s you have 2 kinds, and for the number 2s you have 2 kinds. And the N is going to be 4 divided by 2, or 2, which makes sense because you have N represents the kinds of chromosomes, not the sets. So this 2 right here represents, on the other hand, represents the sets. So for the number 1s, you have white ones and black ones. That's this. But for this N, it's asking about the numbers. So how many kinds do you have? You have the 1 kinds and you have the 2 kinds. So that's why you have 2. The copy number went down to 1, so it went from 2 to 1. Whenever you split a centromere, you go from 2 to 1 because these are identical sister chromatids, right? So this right side is just going to be exactly the same. So the big idea here is that these 2 daughter cells are identical, but not only that, the parent cell is identical with all the daughter cells. These numbers are the same as these numbers, okay? So that's what the numbers enable us to see. Let's go ahead and go to the meiosis, right? This is where it starts getting interesting. The first cell is identical to the first mitosis cell. So let's just copy the numbers. So 2n equals 4, n equals 2, c equals 1. Alright? Now, S phase happens. When S phase happens, by the way, before S phase and G1 phase, This is not what the chromosomes actually look like. I'm just drawing it like figure 8s here, simply because it's more visual that way. So when S phase happens, you double the chromosomes. And, excuse me, you don't double the chromosomes, you double the copy number of the chromosomes, all right? So you make identical duplicates. So now, instead of figure 8, you have the butterflies, right? So the copy number is going to double. The total number of chromosomes is still 4, and the ploidy is still 2. Why is the ploidy 2? Because for number 1s, you have 2 sets. For the number 2s, you have 2 sets. Okay? Don't just look at the colors and see that there are 2 colors. You have to look individually at one kind of chromosome at a time. So 4 divided by 2 is 2, which makes sense because you have 2 kinds of chromosomes. You have 1s and you have 2s, all right? Now, these are homologous pairs. So the 1s are homologous pairs and the 2s are homologous pairs. The black 1 and the black 2 are not homologous pairs. The white 1 and the white 2 are not homologous pairs. You have to be the same kind of chromosomes to be a homologous pair. Okay? Even though you have one from each set. Okay? So these are. Now, here's the key thing. This is not the only way these tetrads, or homologous pairs, could have lined up. These 2 could have switched over, or these ones could have flipped, and the white one could have been on the right, and the black one could have been on the left, all right? So, that's why you have this possibility here. Okay? So, here, it's possible that this one could have been on the right side. This idea is called independent assortment. It happens at metaphase 1 and continues through anaphase 1, but it starts at metaphase 1. All right, and there are other possibilities here too, but I just showed you one. Okay, so in event that this possibility occurs at metaphase, at anaphase and telophase, you're going to split not at a centromere, but you're going to split in between the homologous pairs. That way, since you're not splitting a centromere, you're not going to decrease the copy number. But there's something else you are going to decrease. Let's see what that is. Okay? So in this scenario, you'll have this black one and the white 2 go to the left side, and the opposite go to the right side. Now, this is where things get kind of tricky. People start getting confused here, so listen carefully. Let's analyze both of these cells in terms of these numbers. What's the total number of chromosomes? 2. Here's the hard question. What's the ploidy number? And people make the mistake of looking at this and saying, oh, it's diploid because I see a black one and I see a white one. That's wrong. The reason why it's wrong is because that's not how you count ploidy. You don't just see 2 colors and just call it diploid. No, you have to look at one kind of chromosome and then see how many sets there are for that one kind. So for example, of the number 1s, how many sets are there? Well, there's only one number 1 here. So you can't have 2 sets of it. There's only the black one. Okay? So there's only one copy of number 1 from the dad. Of the number 2s, how many sets do you have? You only have 1 set. You can't have more than 1 set if you don't have more than one number 2. Okay? And it's from the mom. So these are haploid because even though you see 2 colors here, one from the mom and one from the dad, they're different chromosomes. They're different kinds of chromosomes, 1 and 2. You only have one copy of each one. Sorry, one set of each one. So that's why it's haploid. And this is super important, okay? Don't mess that up on the quiz. Now, the same thing with this side. The n number is going to be 2 because 2 divided by 1 is 2. Why does that make sense? Because n represents how many kinds of chromosomes there are. When you go back, how many kinds do you see? You see 2. You see 1s and you see 2s. In humans, you have 23 kinds, so it goes all the way to 23. 1, 2, 3, 4, 5, 6 to 23. But here it's an oversimplified version. The copy number is still 2, okay? Because you see, you still have sister chromatids that are identical. Now, these guys are going to do metaphase 2, right? And anaphase and telophase 2. This time they're going to line up on the centromeres on the metaphase plate. This should remind us of the way metaphase, regular metaphase and mitosis lined up. Okay? So whenever you split a centromere, you're going to decrease the copy number. Okay? So the copy number in these guys is just going to be 1. Right? Because you can see it's just a figure 8 now. Sorry about this, the resolution. How many total chromosomes do you see here? 1, 2. How many sets of chromosomes do you see per type of chromosome? It's still haploid, because for the number 1s, there's only one kind. For the number 2s, there's only one kind. Alright, even if you see 2 different colors here. Now, 2 divided by 1 is 2, which makes sense because there are 2 kinds of chromosomes. Now, we're not talking about the colors, we're talking about the kinds, or the numbers, right? 1 and 2. Now, all of these guys will have the same description here. These numbers apply to all these 4 haploid gametes. Now, there's one thing I forgot to put in here. What is that? Okay? It's that there are actually 2 ways to introduce genetic variation. Only the first one is independent assortment. But the second one occurs earlier than metaphase 1. And it occurs at prophase 1. That's crossing over. Okay? So at prophase 1, the chromosomes cross over. And what that means, for example, if these guys are crossing over, let me just extend this here. Let's say this sister chromatid crossed over with this sister chromatid, and it stole a piece of the black part, and this one stole a piece of the white part, and they swapped. Alright, you'll end up with a number 2 here, with a little black piece down here, and a number 2 here, with a little white piece right there. And so when they split the centromeres, you're going to automatically end up with the little black piece right here going here, but this one doesn't have it. And you'll have a little white piece oh my goodness, I screwed up the numbers here. That's fine. This should be 2, this should be 1. This should be 2, this should be 1. I'm sorry. Okay, and a little white piece right here at the bottom because they crossed over, okay? So as you can see, none of these are identical with each other. They're all different. But they're all still haploid. Okay? Boom, 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 boom. So, now, finally, I'm going to tell you the big idea, alright? The big idea here, I want you to notice, is that it goes from diploid to haploid. Alright, now, this change from diploid to haploid is called the reductional division. And the reductional division occurs in meiosis 1. So this whole thing is meiosis 1, okay? So that's when the reductional division occurs. Now, in meiosis 2, which is from here to here, This is meiosis 2. Is there a reductional division? Well, let's look. It goes from 1N to 1N. It doesn't change the ploidy number. Okay, so there's no reductional division there. No. But there's another thing that happens. The copy number decreases. So C equals 2 to C equals 1. Alright? So the difference between meiosis 1 and 2 is that in meiosis 1, you have the ploidy number gets reduced by half, reductional division, versus meiosis number 2, in which the copy number decreases by half. Right? So let's just zoom out and see the whole thing again. And now that's the whole reason why I wanted you to learn these numbers. Okay. Okay, and everything boils down to the fact that in mitosis you line up along the metaphase plate in a single file line versus lining up in tetrads or homologous pairs. And that makes all the difference when comparing these two processes.